And so as we respond to our community needs, we're driven by needs assessments, it's more and more driven towards prevention. So in a lot of our rural communities, we are essentially a primary source of primary for prevention. And we work across the gamut. So we have direct education, and at the same time, we have county extension agents who are changing the built environment. So we strongly believe in the socio-ecological model that we can tell people to eat their fruits and vegetables, but it's not going to work if they don't have access to those fruits and vegetables. So again, even when you're working with your patient, if you're in a rural setting, look up your county extension office, because they're going to have reoccurring and relevant programming, probably related to the needs of your patient clientele when it comes to nutrition. So that's my plug. Not talking about that today, but I could talk about it all day. And we are, Cooperative Extension, more and more is working with healthcare. So I'll just use the University of Kentucky as an example. We are rolling out the diabetes prevention program with our extension agents, partnering with our diabetes center on campus. So we are actually going to be able to bill for this extension program. And another thing to keep in mind is a lot of our resources, 99% of our resources in our programming is free. So really that eliminates a barrier. So I'm passionate about extension, as you can tell. But we're gonna take a sharp turn. And so the next several minutes, we're gonna kind of look at more of a biological approach to obesity, and specifically visceral adiposity. So we've touched on some environmental aspects. We've talked a lot about the clinical, social, and cultural implications of obesity, but I really want to kind of touch on the biological side of it and what is actually happening in the body, specific with inflammation. So there's more and more literature coming out talking about how inflammation driven, that stems from obesity is really the link between increased body weight and diabetes and heart disease. And so essentially my goal for this talk is for you to kind of understand the morphological and the physiological changes that take place in adipose tissue as a result of obesity, plus maybe be able to discuss some of the cellular mechanisms, specifically cytokine and chemokine production, as well as not just macrophages, we're going to talk about macrophages a lot, but other cell types. And then what does that all mean when it comes to systemic metabolism? That's what we're going to talk about today. And magic. Okay, so we're going to see this image several times today, but I kind of just want to start us out with the basics, talking about adipose tissue under healthy circumstances. So back in the 1940s and for several decades since then, we really thought of adipose tissue as being this inert passive organ. So essentially it's just where we sent all of our energy to be stored until the time of need. But what we've started to see over the past several decades is that is not the case at all. It really is a dynamic, diverse tissue. So if we look at essentially volume of tissue, the adipocytes, or the fat cells here, the yellow ones that have got that big old lipid droplet, they're probably 90% of tissue volume. But if we step back and look at cell type, what we now know is that those adipocytes are actually 20 to 40% of cell composition. So really there's a whole host of other cell types that are present, which suggests to us, even at normal lean conditions, that they are necessary for homeostasis. And so this has been such a blooming field of literature. So all of those other cell types present are considered our stromal vascular fraction, and they include the endothelial cells, the immune cells, specifically macrophages we're going to talk about, fibroblasts, stem cells. We know that adipose tissue is innervated, so we have neurons. So really it is a very, very diverse tissue. I will highlight here that, again, we have macrophages present. So that means even in lean conditions, these macrophages are most likely incredibly important in maintaining tissue homeostasis and cellular stress. So I think this is interesting. 1 gram of adipose tissue is about 1 to 2 million adipocytes, but really all of the other cell types could be as high as 4 to 6 million. So again, it's a very diverse tissue. Okay, so just basic physiology, talking a little bit about what the normal functions of adipose tissue are, because this is an important organ. It's communicating with a lot of other tissues. So what is it doing for us? So if we start just basics, we could have made this list on our own, just sitting here brainstorming. But the physical role of adipose tissue, insulation, so again, those mammals that live in very cold environments. I think the protection aspect is really important. We kind of tell kids, you know, if you fall, it cushions your organs. So it really does protect some of our most delicate organs. So here's an example of the eyeball, where we actually have these small little fat depots that surround it, kind of like a teacup, like packaged, to protect it because it is so delicate. And then cushioning. So think about parts of the body where you have a lot of friction or impact. So the palm of your hand, the bottom of your foot, where you sit, there is more adipose tissue. And so as I was putting the slide deck together, I was kind of laughing because I was like, I could actually list reproduction on this slide because part of selection, some adipose tissue depots, people prefer to be larger. You know where I'm going with that. So essentially, selection, so that could be a part of a physical function of adipose tissue. The one that's so fascinating to me, and a lot of people don't necessarily think of, and this is where I come back to that idea of adipose tissue being really dynamic, is that it is producing a lot of chemicals and molecules that are communicating with a lot of different cell types. So I have auto, para, and endocrine because really it can feed back and communicate with itself. It could be locally, you know, the production of adipokines and anti and pro-inflammatory molecules, which we'll touch on, as well as endocrine. So the adipose tissue is releasing and secreting molecules that are communicating with all parts of the body. And what we know, a perfect example would be leptin, right? So we know that's an adipokine, a hormone that is derived from adipose tissue and communicates with the brain. It's a very, very important axis. Now, if we think about someone that's obese, because we're talking mainly just about regular normal function, someone that's obese, or a person with obesity, there we go, a person with obesity could have body weight where about 50% of their body weight is adipose tissue. So if we're thinking about this as an endocrine organ, think about the profound effects that could have systemically if you have that much tissue producing molecules, pro-inflammatory molecules or adipokines. So that's something that always comes back to me and stands out. Just kind of looking specifically at some of these examples here. Reproduction, we know that adipose tissue provides a lot of the precursors for sex hormones. So that's a consideration. And then I mentioned this, adipokines, and I apologize for the formatting here. So adipokines, I would say probably in the last couple decades, tens of thousands of papers have been released about different adipokines. So these are going to be proteins or hormones that are released directly from adipocytes. So they are derived from adipocytes. And so that gives us this clue that there's a lot of communication going on between our adipose tissue as well as other organs. Now, it's not a one-way, it's not like one walkie-talkie just to one place or whatever. They can talk back because look at all the receptors that adipocytes themselves express. So they're receiving a lot of information and a lot of signals as well from the rest of the body. So again, we have several endocrine hormone receptors that are expressed, just to give us an idea of how intricately embedded our adipose tissue is within our endocrine system, as well as a number of nuclear hormone receptors. And all of these, you know, are involved in regulating gene expression. So again, adipose tissue is busy, and I hope you guys have a new appreciation for adipose tissue. So this is what we always think of when we think about the functions of adipose tissue. You know, it's kind of this master regulator of energy balance because it's the one that's storing and releasing, but it's a little bit more complicated than that. So, you know, the canonical mechanism that we think of going back to the 40s is it's that area where our energy is stored, but also energy utilization. So it's able to recognize when the body needs it. And then we could have a whole conference about the different types of adipose tissue because we know that there's now a color schematic, essentially. So you've got brown and beige and white. But there are some adipose tissues that actually can consume a lot of energy, specifically talking about brown and beige. But I won't get into thermogenesis. Glucose homeostasis, even though it may play a small role in being a glucose sink itself, it does take up some glucose. But really, it's those molecules that it's releasing, those inflammatory molecules or those adipokines that really are involved in making sure that, that our body is able to maintain glucose homeostasis. And then last is lipid homeostasis. So it is 100% managing our lipid. It is storing it. It's also providing it if it's necessary for the other functions fatty acids may have. So thinking about cell membrane development or precursors for other molecules. So again, the lipid homeostasis aspect is really important too. So it does a lot more than just storing our energy. So I'm specifically talking about abdominal or visceral adiposity. And so I kind of just wanted to point out the differences. So you guys probably already know this. But we do know that our visceral or central adiposity has much more deleterious effects on the body than the subcutaneous. And so here, subcutaneous, gluteal and femoral, going back to that reproduction. Some people like larger adipose depots. But we're really interested in the visceral, and in humans, that's going to be mainly in the form of the mesenteric and the omental being the largest here. And so it's interesting because these more central adipose tissue depots, they actually have different metabolic rates. So they have increased lipolysis or the production of more inflammatory markers. And that's most likely such an issue because it has direct access to the liver. So that's something to keep in mind. And maybe one of the mechanisms or reasons why this specific adipose tissue depot is so much more dangerous, essentially, than others. And an important thing that I want to point out, because pretty much all the studies that I'm going to show you and the data moving forward are from rodents because that's where the majority of this work is done. So how does that translate to humans? Because I think this is something that's kind of lost in the translation when we're looking at the literature. So as I mentioned, the central adiposity that in humans that seems to be most deleterious is really the omental and the mesenteric. So you can see it's right here just situated around our central organs. But in a rodent, when we look at the specific mechanisms or the role of a specific cell, a lot of times we're actually sampling and utilizing this gonadal adipose tissue or this perigonadal adipose tissue. Now, in humans, that's almost absent, essentially. And so this omental and mesenteric adipose tissue, it's almost absent if there's any at all in rodents. So just kind of keep that in mind, that we're essentially using, again, it is centrally located in the rodent, but it's this perigonadal. So what you'll see going forward is maybe epididymal adipose tissue, and that's what that's a reference of used from a male mouse. But that is not necessarily present in humans. So a caveat. All right, so we saw this lovely image, and I'm assuming you know what's going to pop up on this side, because I'm going to show you how adipose tissue changes under obese conditions. But here when we look at our lean phenotype, we essentially see just everyone living in harmony here. Our adipocytes are uniformly shaped. They're able to manage the lipid that's coming on board, whether that's from the liver or if it's from the diet. And then we also have adequate vascularization. So these are living cells. Adipose tissue looks white because there's low vascularization, but at the same time there are capillaries that are providing necessary oxygen for cellular respiration. So under obese conditions, here we go, they're all angry, everyone's mad. So we have our swollen adipocytes, which is considered hypertrophy. So these cells are trying to expand and accommodate and sequester all of that excess energy. And we see this remodeling that takes place of adipose tissue. So as those cells swell, and we need them to have some degree of plasticity to be able to grow, what happens is that we start to have stress on the extracellular matrix. So that's going to cause some cellular stress. So those cells are not going to be able to be as plastic as they need to be. And at the same time, we actually see increases in fibrosis. So those fibroblasts under stress start to lay down collagen. And so again, that puts more stress on that cell as it's trying to accommodate and essentially prevent that excess lipid from being toxic. Other issues, so that's more kind of a physical stress, but other issues actually may be intracellular. So we have stress on the endoplasmic reticulum. And so that's going to induce some inflammatory pathways within the cell as well. And then the last one is hypoxia. So I mentioned that our tissue was originally adequately vascularized. But what we tend to see is that some cells will grow larger and swell, will lose that uniform shape. And that means some cells may be pushed farther away from their oxygen source. And so those conditions tend to become hypoxic. So we have need for oxygen, but we're not able to meet that need because it's being consumed too readily. So again, a lot of things are going on that are kind of promoting inflammation. Another thing that I want to point out is that we have some cells up here that we didn't originally have. And so you can see that our original resident macrophages are still present, but what tends to happen is through all of these different mechanisms, of pro-inflammatory mechanisms, these guys kind of switch from their happy, harmonious, anti-inflammatory phenotype to a more inflammatory phenotype. So they were originally releasing anti-inflammatory molecules, now they're releasing pro-inflammatory. At the same time, we've got both these guys and our adipocytes releasing cytokines and chemokines, and we'll talk a little bit more about those in a minute. And so they're actually recruiting additional macrophages from the systemic circulation. So not only have we made our resident macrophages angry, we've also sounded the alarm, and we've asked everyone else to come visit as well. So you can kind of see how I'm setting this up to show this chronicity of inflammation that we see under obese conditions. So here, if we just look at some histology, if you like to see that, this is actually adipose tissue from, again, periogonadal in a male rodent, but very similar to what we would normally see. So we have uniformly shaped adipocytes. They're able to manage the lipid appropriately. We don't see a lot of fibrosis. We don't see a ton of other cell types present here, actually, but there you go. So we can see that very swollen adipocytes. We have some that are much smaller. And then look at all this fibrosis and the additional cells that are either activated or been recruited. So here is exactly what we would see, especially with the increase in the extracellular matrix. All right, so this is kind of setting us up for the rest of the talk and what I'm going to go through. So this idea of immunometabolism, it isn't really new, but it's something that we're talking more and more about when it comes to metabolic dysfunction under obese conditions, essentially. So it's looking at how does the immune system itself, how is it involved in metabolic regulation? Is it itself a regulator? Is it the immune response actually a regulator itself of how our body is able to metabolize and utilize energy? And so we're going to look at this from the lens of the adipocyte. So we're adipose-centric. You know, we talked about liver-centric. I'm actually going to take the stance today of being adipose tissue-centric, and specifically the adipocyte, because I think it's lost a lot of times in the role that it actually has in promoting inflammation. So we briefly already touched on several of these, and I'm going to just show you a little bit more data to support that. And then I hope to make the connection of how that truly drives the activation of those resident macrophages, as well as the attraction of new macrophages to the tissue. And these are some of the specific mechanisms. So chemokine and cytokine production, and then also the role of other immune cell types. So not only are we talking about macrophages here, we're also going to talk about the adaptive immune system. So we know both arms of innate and adaptive play some role when it comes to inflammation associated with obesity. And then lastly, just connecting these mechanisms to the big picture, what the patient is actually experiencing. So insulin resistance, glucose intolerance, and dyslipidemia. And I'm going to say that you could have a whole conference on just this slide and all the different talk on everything up here. So I'm going to just give you some snippets of what the literature says, and we're taking a very broad approach for something that is very minute and detailed. All right, So let's start off looking at just a few interesting studies when it comes to triggering the adipocyte when it comes to inflammation. So for some of you all, do not get overwhelmed by this slide. I know it can, I just wanted to draw your attention to a couple things up here. So we're actually just looking at an adipocyte here. So here's the cell membrane. And I'm going to kind of share with you a little bit about how specifically saturated fatty acids, palmitate, so C16, really serves as a stimulus for these adipocyte-specific inflammatory pathways. So here we've got our saturated fatty acids, most likely palmitate, which is derived from either lipolysis or de novo lipogenesis. And essentially, they can work both indirectly and directly on the adipocyte to stimulate cytokine production. So here, if we look over on the right, we have our palmitate, which can actually enter into the cell and then ultimately inhibit insulin sensitivity, as well as contribute to the production of inflammation and the gene expression of cytokines. What's interesting that we're just now realizing is that palmitate, or these saturated fatty acids, which are very toxic, can actually bind to the TLR4 receptor, or the toll-like receptor 4. And so usually this is a receptor for cytokines or pro-inflammatory molecules. So what we see once that binding occurs, we actually have the promotion of cytokine production as well, so both indirectly and directly. Now, I don't want you to have to pay attention to anything else. But what's interesting is that if we specifically knock out this toll-like receptor 4 in adipocytes only, we actually see that fewer macrophages are recruited into the adipose tissue. So this is our control, and just a couple different adipose tissue depots here. But what you can see indicated by the red staining, the MAC2 staining, which is a very common marker of macrophages, is that we have quite a bit of red expression. So even in our control, we have a lot. So in the control, it does have our receptor. But if we knock out the receptor, we see that the amount of macrophages is drastically reduced. So again, it's the adipocyte itself that is largely contributing to macrophage recruitment. So I mentioned hypoxia. So again, this is the idea of an imbalance in oxygen needs versus oxygen consumption. And it's a strong driver of inflammation in all tissues. But if we look here at some adipose tissue stained from mice, again, for essentially this adduct of hypoxia, we see that even with just a 3-day high-fat diet challenge, we already start to see hypoxia within our cells. And so why in the world is this an issue so early on, right? Because we haven't even had time for the cells to swell and to push others away physically from their oxygen source. So what could be driving this? And look, it's this guy again, saturated fatty acids, so that toxic palmitate, essentially. But these free fatty acids, it's speculated, are increasing the respiratory needs of the adipocyte itself. So we know free fatty acids are going to drive the mitochondria to want to work harder, but if the oxygen isn't present, then that's an issue. We have an imbalance there between the oxygen needs and what's the oxygen that's being consumed. So this is a cell culture study, essentially, where we took an immortalized adipocyte cell line, and this is our control. Again, here's our hypoxia marker, this adduct, so we don't really see much green positive staining. But if we just treat our cells for 6 hours with free fatty acid, specifically palmitate, look at this robust increase in hypoxia that we see very quickly. Very quickly. So hypoxia may be one of those first initiating factors. It's not known yet, but it may be one of those first initiating factors that's starting to stimulate or trigger the adipocyte to respond with its inflammatory mechanisms. I mentioned the endoplasmic reticulum stress. So again, intracellular pro-inflammatory pathways are activated. We start to see the expression again of more cytokines. When I teach my students in biochemistry the idea of inflammation and obesity, they're like, well, they just lose track of all the cytokines, essentially. They're like, who's secreting the cytokine? What does it do? Literally, it's just a cycle. It's the adipocyte and the macrophage responding. So here again, we see more cytokines being produced. And then you might start to see how this connects to some of the greater systemic issues that are driven by adipose tissue-derived inflammation. So ER stress actually is linked to reduced expression of the insulin receptor. So we know insulin sensitivity on adipose tissue is incredibly important in maintaining glucose homeostasis, as well as lipid management and lipid homeostasis. And then once ER is stressed, it's actually going to cause lipolysis to become unregulated. So again, two of these major mechanisms that are driving a lot of the other metabolic dysfunctions we see. Fibrosis, so you know that rigidity that we start to see in adipose tissue under obese conditions, specifically collagen 6. So to kind of explain a little bit about what you see, so your wild type is going to be our control, and then the middle picture here is essentially a slide of adipose tissue from a rodent that has a genetic form of obesity, which is OB, and that is a mutation in the leptin gene, so they do not have functioning leptin. So even without a high-fat diet, we see that there are very large, non-uniform adipocytes. But what's interesting is if we cross this genetically obese mouse with a mouse that is deficient of collagen 6, we see that the adipocytes are even larger. But what's interesting is that we look metabolically at those mice. They are insulin sensitive, and they have less inflammation. So essentially, the adipocytes are able to grow and accommodate all that excess lipid, and yet they're fat. They are obese mice, but yet metabolically they are healthy. So that kind of speaks to maybe this is a little bit more important than we realize, specifically fibrosis. And then a few other pieces of data from that study, essentially. F480 is a very common marker of macrophages, and then MCP1 is a very robust chemokine that's expressed under obese conditions, and we'll talk about that in a second. But what we see in these mice that are essentially deficient of collagen but are genetically obese, they even have reductions in macrophage present in their adipose tissue, as well as reduced production of chemokines, which are implicated in obesity as well. All right, so again, that was kind of talking about some of those specific mechanisms to the adipocyte. So what is that doing to the other cells that are around it? So here's where I want to talk about cytokines, chemokines, and all the other immune cells that are involved. So first I want to talk about chemokines. So chemokines are essentially molecules that are released that are involved in chemotaxis of immune cells or macrophages. So they work on a gradient, so the stronger the concentration is, the more likely it's going to be able to pull or recruit essentially another cell type to come to that area locally. So the one that probably the most work has been done on in obesity is MCP1. And so that's the one I want to focus on here. So essentially what we have on the X here is several different adipose tissue depots. So EP would be the epididymal that we talked about, retroperitoneal, mesenteric, and subcutaneous. And after essentially 3-month high-fat diet for our mice, if you're not familiar with this work, we literally put our little mice on high-fat diets. It's about 60% of their kilocalories come from fat on this diet. It's really greasy, it's gross. And it's supposed to mimic a Western diet, essentially. So after about 3 months, these mice are obese. And what we see if we look at the tissue is that MCP1 production in all of the different adipose tissue depots is drastically increased. So we know that this adipose tissue is laying down and producing a lot of this chemoattractant. But what's interesting, and some additional studies have shown, as I mentioned earlier, there are a lot of different cell types in adipose tissue. So who is it that's actually producing this guy? Who's responsible? And so this study is interesting because they actually isolated adipocytes specifically from epididymal adipose tissue 3 days after a diet challenge and 1 week after a diet challenge. And so those adipocytes themselves, looking at the amount of MCP1, we actually already see a significant increase in this chemoattractant being produced specifically by the adipocyte. So again, the adipocyte is playing a role in recruiting those additional macrophages into the adipose tissue. Now I have to show you this because this is a figure that was probably, it's out of one of the seminal papers when it comes to telling us for the first time that macrophages play a role in adipose tissue. So this is back in 2003 from Anthony Ferrante's group. And essentially what it's telling us is that the adipocyte area serves as a really strong predictor of macrophages being present in the adipose tissue. So as the adipocyte area becomes larger, we actually see an increase, again, this is that macrophage marker, a significant increase in the number of cells that are positive for the macrophage marker. Very strong correlation. And what we now know is that the presence of macrophages in adipose tissue directly correlates with BMI. So the more adipose tissue you have, the more macrophages you have. So again, this goes back to the idea of a person with obesity. If they have more and more adipose tissue, think about the inflammatory response and the number of immune cells that they have activated in all of their adipose tissue. So again, this is kind of just a visual representation of what I just said. So this is from rodents, so keep that in mind. But we have essentially where we've got staining for that F480 that I mentioned a minute ago, that macrophage marker. So here's our control. Even after just 3 days of a high-fat diet, we start to see the formation of these things we call crown-like structures. So that's an indication of macrophages being present. So here's 1 week, this is quite a bit, 2 weeks, 5 weeks, and 10 weeks. And so even after just a couple months, 2 to 3 months, we can see that there's a robust increase in the macrophages that are present in this adipose tissue. Now I'm going to speculate that probably some of these earlier responses we see is essentially the activation of those resident adipose tissue macrophages. And what we see here most likely is certainly the recruitment of additional macrophages to the tissue because of that, the chemokines and the cytokines being produced. All right. So some of the other really important cytokines that have been implicated in obesity-associated inflammation are IL-1 beta as well as TNF-alpha. And you may be very familiar with some of those. What we tend to see is that both of these are dramatically increased in adipose tissue as well as circulation under obese conditions. And this has been verified in both rodents and humans. So this is something that we really do know. So here, probably the worst culprit here is the TNF-alpha. And so as you can see here, it increases under obese conditions. What we see over here on the right, it's from 1993, so it's pretty old, but really I'm showing this because it is the first time that we actually showed that adipocytes themselves were responsible for the production of this cytokine that has its implications in a number of health conditions. So essentially what we have are lanes 1 and 2 are adipocyte fractions, and then lanes 3 and 4 are going to be the fractions that include all of those other cell types that are present in adipose tissue. And then the odd numbered are lean or control, and then 2 and 4 are high fat. So you can see under the high fat conditions, the adipocytes are the ones that are primarily responsible for pushing out this TNF-alpha, which is really driving both adipose-specific problems as well as systemic problems. And that there is some production of TNF-alpha in our stromovascular fraction, most likely from our adipocytes or from our macrophages, but it is nowhere near to the degree of the adipocytes themselves. So again, just kind of emphasizing how the adipocytes themselves are the ones that are contributing to the chronicity of this issue. So this is kind of a schematic picture to put this, to step back for just a second and look at all the things that we've talked about so far. So essentially we have our adipocytes who become angry and inflamed, and so what they start to do is send out their distress signal, which is naturally the production of some of these inflammatory markers to call in reinforcement and help. What tends to happen is this is a very robust response, unnecessary response, for what it actually needs for the time. So we see the production of MCP1, which is that chemoattractant, so it's going to start to recruit those other macrophages. We see robust TNF-alpha production. We know that With insulin resistance, lipolysis is unregulated, so we see an increased release in palmitate or saturated fatty acids. This is going to promote either the switch of our happy anti-inflammatory macrophages to pro, or the recruitment of additional, and then we start to see, we're going to talk about this mechanism in a second, we start to see the production again of more pro-inflammatory molecules. Again, TNF-alpha is produced, IL-1 beta, which we just mentioned, and it ultimately kind of feeds back. So you can kind of see this crosstalk between the adipocyte and the immune system itself. All right, so I touched on this already, that within that central adiposity, especially under obese conditions, we start to see this switch from our anti to our pro-inflammatory. If you're working within the literature or within research, a lot of times these are identified by CD11C+, which is just a cell surface marker, and that's an indication that it is a pro-inflammatory macrophage. And I alluded to this whenever I was talking about adipocyte size and F480, but really we do know that the presence of inflammatory macrophages in adipose tissue directly correlates with BMI and insulin resistance. So this is, again, something that is consistent between both rodent studies and what we see in humans. All right, so I briefly want to touch on all the other cells that are involved in both the innate and the adaptive immune system. So first of all, neutrophils, natural killer cells, mast cells, we know that they're implicated in some way in adipose-derived inflammation. All of these are increased in presence in adipose tissue under obese conditions. The one that's really interesting to me specifically are mast cells, because what we've seen in some rodent studies is that if mice are deficient of mast cells, they are 100% protected from diet-induced obesity. It's like, what? How does that even work? We don't know, but that is just something that kind of speaks to they are most likely more involved than we even know or realize at this point when it comes to the literature. And then when we look at our adaptive immune system, I think this is really interesting, and this is an area that research is starting to take off. We haven't exactly pinpointed the temporal organization of immune cell response in adipose tissue under obese conditions. So we don't know if it's the macrophages that are really first, but there are actually some studies that suggest that cells from the adaptive immune system are actually infiltrating the adipose tissue before those recruited macrophages. Don't even know what really that means, but it's interesting to keep in mind. When it comes to T cells, we know that both T cells and B cells significantly increase in obese adipose tissue. But what's something important to note here is the reduction in Treg cells. So those are kind of the good guys when it comes to immunosuppressive capacity. And so what we tend to see in adipose tissue is that the abundance of Treg cells specifically decreases. So if that's the case, we actually lose essentially the immunosuppressive part of the immune system, which may speak to why we have such a robust inflammatory response if those guys aren't around and helping out. And then lastly, B lymphocytes also increase specifically in the visceral or central adipose tissue in mice. Okay, so that was a lot. I apologize. Let's zoom back out for a second. So you know on the other side, the next slide is going to be the angry picture. But here's the picture of essentially lean conditions. This is what we know and what we've already talked about. So our adipocytes are small. They're able to manage this lipid. We have the presence of some of these good guys. So we have our anti-inflammatory macrophages. We've got our Tregs over here that are kind of helping. We have the release of anti-inflammatory cytokines. It all is right in the world regarding tissue maintenance and metabolic health. The angry one. So here we've got chronic excess nutrient that our adipocytes are having difficulty managing and onloading or onboarding. And then we start to see the increase in all of these other cell types that are contributing to this pro-inflammatory phenotype. So we've got our switch from our anti to our pro-inflammatory macrophages. We've got our B cells. We have the production of these pro-inflammatory cytokines we talked about, IL-1 beta and TNF-alpha specifically. And then not only are we going to have tissue dysfunction at this point, but when things kind of get really out of control, that's when we start to see more of those systemic effects. And so that kind of is what leads us to, for the last part, last 10 minutes talking about how all of this manifests essentially into systemic metabolic dysfunction. All right, so I mentioned this pathway earlier that we'd see it again. So the NF-kappa B kinase complex. I'm not going to get into the details, but this complex is a bad guy, essentially. And it's what largely contributes to the production of cytokine gene expression. So it's stimulated largely by TNF-alpha. So we've already talked about that quite a bit, and so we know that our adipocytes decide under obese conditions to start producing a lot of that. So what we're going to see is the activation of this pathway in adipose tissue within obesity. And so these are some of the implications of that pathway when it's turned on. So obviously we're going to have increased cytokine production. What we also see is that GLUT4, so the receptor that's going to be taking up that glucose, that little bit of glucose out of the bloodstream in our adipose tissue, that expression is going to go down, so that's not going to be happening as much. And then the last one that's really problematic is it hinders the insulin receptor signaling. So it's going to contribute to insulin resistance in our adipose tissue. I wanted to show you essentially why this guy is so bad, because these are just examples of all the target gene products of this complex when it's turned on that have implications in insulin resistance, diabetes, and atherosclerosis. So we've talked about a few of these, and maybe if you do research or clinical work, then some of these other ones look familiar. So really turning this guy on is very detrimental to the cell when it's producing and regulating these specific genes. The other specific pathway that is largely connected to more systemic function is the J and K pathway. So you may have seen that a couple slides back when I showed that schematic of how we have this chronicity issue with adipose tissue inflammation. And again, once this guy's turned on, it's regulating gene expression, essentially, and it's contributing to the cytokines that are being produced, so specifically IL-1 beta and TNF-alpha, IL-6, you may have heard of that. And then it's also impairing the function of insulin receptor. So again, when both of these guys are turned on, we're really going to have issues with the adipose tissue responding to insulin. All right, and so this is just a kind of a visual talking about how all of this inflammatory cell signaling is impeding the adipocyte's ability to do its main job of lipid management. And so we're focusing specifically again on this really bad guy, TNF-alpha. So first of all, it's going to reduce the ability of the adipocyte to make triglyceride, so to put that fat into the stored form. And it's doing that by essentially blocking gene expression and impeding the ability of insulin to signal. And so I always teach my undergrad students, insulin is that storage hormone, so it's going to promote the storage of things. So if insulin is signaling appropriately, that adipocyte's going to be making triglyceride. And so in this case, if this is impeded, then we're not going to have the production of triglyceride like we need. The other one down here, TNF-alpha, bad guy, it's increasing lipolysis. So it's actually going to block perilipin, so it's the proteins that line that lipid droplet, and it's going to promote lipolysis directly on hormone-sensitive lipase. So we're blocking the storage, and we're promoting lipolysis, so we're going to be pushing free fatty acids out of the cell when we don't need that. And so again, that's going to lead to increased circulating free fatty acids. It's also impeding the ability of lipoproteins to drop off lipid to the cell as well. So again, just this one guy alone is regulating several important mechanisms when it comes to lipid management on the adipocyte. And so again, getting a little bit more big picture, that chronic nutrient excess and the inability to manage all of that lipid that's coming in is where we start to see the accumulation of lipid in other metabolic tissues. So here, we know this visceral fat is detrimental, but then once we have issues with the liver, we have major issues that are going on. And then we also see the accumulation of ectopic fat in the pancreas, the kidneys, muscle, and the heart. All right, so just a few more slides here. So just looking at what essentially is happening between that transition from lean to obese, we're seeing the increase in pro-inflammatory molecules that are signaling both to the adipocyte itself, within the adipose tissue, and then systemically as well. A lot of those are pro-atherogenic, and then we also see that poor lipid mismanagement, so that's going to lead to some lipotoxicity. And so essentially those all manifest in what the worst comorbidities of obesity are: insulin resistance, the ability of the pancreas to appropriately respond to glucose, and then finally, and what gets everyone is heart disease, atherosclerosis. So here we've covered a lot, but I hope that you kind of have a better understanding of what the role of the adipocyte actually is when it comes to inflammation under obese conditions. And so you can kind of see how it's leading to the activation and attraction of not only macrophages, but other immune cells as well, and how this truly manifests in insulin resistance and lipotoxicity within the body. And so I want to kind of wrap up with this last question. So is there therapeutic potential targeting adipose tissue inflammation? So this is probably something you guys are interested in. So there have been a number of studies that have shown that caloric restriction and weight loss actually do significantly reduce inflammation, and that is tied to improvements in some of those comorbidities like insulin resistance. But where does that go? What does that go? Is there an approach when it comes to drugs and food? So there have been a small number of clinical studies looking at pharmacological approaches to reduce adipose tissue inflammation. Now, there have been some success, but those studies have been very small, or they're with a very specific population. So that makes me want to question a little bit more about what about food? How can we use food as a tool to really combat the inflammation that we see when it comes to obesity? That's a question for you to ponder. This is a place that the literature hasn't really gone yet, and I think it needs to go. And so you guys, having kind of attending this conference and showing interest in this type of work, you are individuals that could kind of take this question and run with it and help to find an answer for it.